So our next presenter will be Dr. Angie Peltier with the University of Minnesota. She'll be talking to us about soybean cyst nematode update. And Angie is based out of Crookston, Minnesota, and doing great work up here. So Angie, when you're ready, you can present.
Okay, can you see and hear me then just fine?
Yes, I can. And anyone else can comment anytime.
Okay. So David Key asked me to talk about soybean cyst nematode and some of the research that the Soybean Research and Promotion Council has funded. And so when David asks, I deliver. So that's what I'm talking about today. I'll share with you whose work it actually is because it's not my work. But before we get started, I just want to give you a quick review of some of the concepts we're going to be talking about in this presentation. As you know, most of you, SCN stands for soybean cyst nematode. It's a little worm that lives in the soil and it parasitizes roots of soybean and dry bean. If you look at that top picture on the right, that arrow, the tip of the arrow is actually pointing at what is a cyst of SCN. Cysts are dead female nematodes filled with eggs. So that's what soybean cyst nematode looks like. Above that arrow, that just for scale reasons, that's one of those nitrogen fixing nodules that Missy was just talking about. The more eggs in a given volume of soil, the greater the chance for soil yield, or excuse me, soybean yield loss. Let's also talk about iron deficiency chlorosis, and that's the picture on the left right there. So plants need iron to make chlorophyll. And if you'll recall from your biology classes, chlorophyll is that green pigment that's needed for photosynthesis. And without photosynthesis, plants would not be able to grow and develop. And
Angie,
yes?
Could I interrupt you 1 second? We want to see your screen that you're sharing, or you need to share your screen, or do you have one that you're showing us?
Oh yeah, I'm sorry.
Rest of the quality is excellent. So just if you have a chance to share the screen.
Oh yeah, I thought I did, but I must not have. Okay, yep. So I'll start from current slide.
Perfect.
Now you'll see.
Yeah, if you want to just start over very quickly, everyone's patient at home here. It's going well, but now we can see your screen.
Now you see my pictures.
Yep.
So here, let me change the display too. So can you see a full screen of
Yes.
picture? Okay.
Yep, it looks excellent. So just take off. You're good.
Okay, yeah. So up above on the right, that arrow's pointing directly at a cyst of soybean cyst nematode, and right above the arrow, just for scale purposes, that's that root nodule that Missy was just talking about that fixes nitrogen in soybean roots. IDC, that's that picture on the left there. Essentially, like I was saying before, plants need iron in order to produce chlorophyll, and that's that green pigment that helps plants to photosynthesize. And without chlorophyll, plants don't grow and develop normally and eventually will die. Most of our soils have plenty of iron, but it's not necessarily in a form that our plants can use, our soybean plants can use. And so what happens is soybeans will tend to release organic acids into the soil, and that solubilizes iron into a form that can be carried with water up and into the plant. In our soils, unfortunately, many soils in Minnesota, North Dakota, Iowa, we have soils with high calcium carbonate content. And what happens in those soils is that plants will release those organic acids, but those alkaline pH soils with a high calcium carbonate content tend to neutralize those acids. So they're not able to work on that iron and make it into a form that the plants can take up. So when plants aren't able to meet their iron needs, to to make that chlorophyll, the tissue between the leaf veins turns that yellow color. And chlorosis is just a fancy term for yellow. Also, we'll talk a little bit about mycorrhizal fungi. These are fungi that live in the soil, and they form relationships with our plant roots. So you see that diagram on the right there, those boxes, those little brick structures, those approximate plant cells. And then those orange threads, that's mycorrhizal fungi in a mycorrhizal fungus inside of a plant root. So what happens is they infiltrate the plant root, and then they spread a series of threads throughout the soil, essentially increasing the range of plant roots. So they're able to actually mine more of the soil than plant roots alone can do for some of the water and mineral nutrients that the plants need. Up here, most of our crop plants do form these mycorrhizal fungi relationships, with notable exceptions of sugar beet and brassicas like canola. So a couple of years back, some researchers were doing work looking at crop, basically the crop that preceded soybean and how that preceding crop might affect IDC in soybean. And what they found is that when soybeans were grown after corn, there were significant IDC symptoms, but when soybeans were grown after sugar beet and canola, there were few, if any, IDC symptoms. And so this led them to the hypothesis that there might be a relationship between IDC and mycorrhizal fungi. So a series of experiments was set up to look at the interaction among these different factors. So experiments were set up in a greenhouse in pots with different SCN population density at planting time. So a 0 SCN, a low, and then a high population density. The low density was 500 eggs in 100 cubic centimeters of soil. The high density is 10,000 eggs in 100 cubic centimeters of soil. 3 different soil pHs, so slightly acidic, 5.6, pretty neutral, 6.9, and then slightly alkaline, 8. And then with or without mycorrhizal fungi. So they looked at those different treatments and how they affected soybean plant biomass, or the dry weight of the plants grown in these conditions, leaf chlorophyll content, which is a way of objectively measuring IDC symptoms, and then SCN population density over time. So after enough time had elapsed for 1 or 2 additional SCN generations to occur. In the interest of time, with 15 minutes devoted to this presentation, I'm gonna skip the mycorrhizal fungi portion and just stick to the SCN and soil pH. So these are the 2 gentlemen. So University of Minnesota plant pathologist, nematologist, Dr. Senyu Chen, and then Dr. Jim Curley, both Jim Curley's at the main campus in St. Paul, and Dr. Chen's down at Waseca at a research center. Now that citation down at the bottom, this work has been submitted for review in the Phytobiomes Journal, and with the hopes that they can get that work published. Know that, again, this research was funded in part by the Research and Promotion Council in Minnesota. So here is a graph showing the effect of soil pH and SCN status on plant biomass. So on the left is soybean biomass 35 days after planting, and on the right, 65 days after planting. Those numbers below the sets of 3 bars, that's the soil pH. So 5.6, 6.9, and 8. And then the different patterns on the bars indicate the different SCN status. So the polka dotted bars are plants that were grown in soil that had no SCN. The striped bars were that low population density of SCN, so 500 eggs. And then the gray bars were 10,000 eggs per 100 cc's. So what you can see is different letters above the bars. So one more thing, different letters above the bars mean that for those 3 bar sets that those treatments are significantly different. So for example, in 35 days after planting on the left, at a pH of 6.9, the 0 and 500 eggs SCN treatments were statistically similar, whereas that 10,000 eggs treatment was significantly lower biomass-wise from the other 2 treatments. So results-wise, soybeans grow best at mildly acidic soils. And so it's not really a surprise that when there's no SCN, so that's those polka dotted bars, if you look all the way across, soybeans grow a little bit better at a pH of 5.6 and 6.9 when compared to a pH of 8. And that's regardless of SCN status. Biomass at that low level of SCN infestation, so those are those striped bars, maybe there was a little bit of a trend at that 65 days after planting, a trend with pH where the higher the pH, the lower the biomass with a little bit of SCN, but you really have to struggle to see that. So at that high rate of SCN though, that is where we had significantly reduced total plant dry weight when compared to both the low and the no SCN treatments. It also looks as if plant growth was a bit more retarded in soils with a pH of 8 with that high SCN initial egg count. Do know that that high egg count treatment at 10,000 eggs in 100 cc's of soil, we would expect some yield loss to occur even when planting a soybean, an SCN resistant soybean variety. Here's the effect of soil pH on final SCN egg population counts. So on the left is 35 days after planting. On the right, 65 days after planting. And the different bars here, the different colors, indicate different soil pH. And so those maroon bars, that's the pH 5.6 treatment, red 6.9, and yellow is 8.0. You can see that at 35 days after planting, this would be enough time for SCN to have these eggs to have hatched, to have infested or infected the soybean roots, and then form another generation. At 65 days after planting, they could have had 2 cycles of reproduction take place in the pots. So you can see that at 35 days after planting, the highest SCN population densities were in pots that grew plants at a pH of 8. At 65 days after planting, the numerically highest SCN population density was at a pH of 6.9. And the authors, the researchers really speculate here that maybe plants just grew that much better at that pH 6.9 that they were able to better support SCN reproduction at that pH. But do know that there was not a statistical difference between the 6.9 and 8 pH soils. So like I said, they also looked at the effect of soil pH and greenness or yellowness of the plants. And they used, rather than relying on visual cues, subjective visual ratings, they decided to use a chlorophyll meter. At 5 through 9 weeks after planting. So each week they went in and they could just take and use this little meter and kind of squeeze soybean leaflet tissue and read it without, take their reading without having to destroy the plants. And again, those different color bars are the different pH, soil pH. Each week, plants grown at a pH of 8 were statistically less green than those grown at 5.6, and in some instances, than plants grown at a pH of 6.9. And if you look at weeks 7 through 9, plants were statistically less green than when grown at a pH of 6.9 than when grown at a pH of 5.6, which makes sense. Like I said, soybeans grow best in a slightly acidic soil. But when we take a look just at the numerical trends over time, chlorophyll content readings indicated that plants grown at a pH of 5.6 tended to get greener over time. So they were accumulating more chlorophyll over time. And you can see the opposite trends for plants grown at a pH of 6.9 and 8.0, where over time, greenness tended to numerically decrease. Here's a picture. So this, unfortunately, will be my only pictures of plants in this presentation. These are soybeans. You know, if we were together, Chris, I'd be quizzing you and everybody else in the room, asking you what you thought the difference was between these two plants. I will give you a hint. Neither one of these two plants were grown with SCN. Here's some of their counterparts. So these plants were grown with SCN in the soil. So these were grown with that high level of SCN at 10,000 eggs per 100 cubic centimeters of soil. And you can probably imagine that the main difference between these two sets of pictures is soil pH, and you would be right. So on the left are plants that were grown at a soil pH of 5.6. You can see the effect of having SCN in that environment on plant growth and development. So the plants are smaller, and they tended to be a little bit more yellow. On the right, those were plants grown at that higher, more alkaline pH. You can see that plants are significantly visually more yellow when they had SCN in the soil, infecting the plants. And so that really kind of helps us to visualize what was going on in these experiments. And that red background sure helps us see those colors too. So in conclusion, it should, I guess, come as no surprise to most of us that soybeans grow better at those acidic soil pHs rather than the alkaline pH. And when compared to growing soybeans with no SCN in the soil, SCN, even at that low population density, results in lower biomass. And we see a trend that accelerates with higher densities, at higher SCN densities and a higher soil pH, we tend to see much less soybean biomass with both of those factors being present. So one thing to keep in mind is we knew researchers and research since I think it was the 90s has shown that in both Iowa and Wisconsin, that soil pH has a relationship there's a relationship between soil pH and SCN reproduction. And so we always knew that when you have high soil pH or more alkaline soil pH, that you tend to have higher SCN population densities. And these densities are much harder to drop over time, even with intensive management in high soil pH. And so we didn't really know whether it was simply a fact that maybe SCN survived better in alkaline soils. What this research showed is that SCN, at least at those first 2 generations, is just able to reproduce better and produce more eggs during those first 2 generations in the more alkaline soils. And so all of this together really just goes to show that we really need to actively manage both IDC and SCN. And for IDC, you know, taking advantage of those great NDSU IDC variety ratings is wonderful. It's a great resource to have. And then consider targeting soybeans in those areas that always have IDC with some of those in-furrow iron chelate products. Active SCN management is also really vital. So we need to sample for SCN, 1, to determine whether our fields are infested, because there are still some fields where we just don't know our status. And then 2, to see if we're able to keep those population densities low, because that's our goal. We'll never get rid of SCN, but once it's in a field, but keeping those densities as low as possible is going to be our goal. SCN management, our best tools still include rotating away from our host crops of SCN to one of the numerous crops that we can grow and can grow well up in Northwest Minnesota. We also need to rotate, if at all possible, our sources of SCN varietal resistance, because we all know what happens when we rely on just one herbicide for managing weeds. We end up with a herbicide resistant weed issue. That same thing can happen when it comes to SCN populations and a single source of SCN resistance. And so break up our varieties with different sources of resistance. We can slow down that acceleration towards SCN populations that are able to resist the resistance, if that makes sense. So I'm not sure where I am on time or if there's time for questions.
Yep. So Angie, you're doing very well. We do have a few questions here. So I'll just ask the question. So one, Greg LaPlante was wondering, when you show your different pH soils and how the plants grow, is that soil low in salts and carbonates, but just higher pH?
Shoot, I think I forgot my paper. The main difference in that soil is it's not only a pH issue. I'm not sure if they figured out the carbonates content in that soil. I do know that they actually tested the soil there for iron content, and so it had a lower iron content too.
Okay.
That high soil pH, but if somebody wants to put their email address or just give me, here's my contact information, I can send you the exact information about the 3 soil types.
Yep. And when you add iron in there, I think we're almost talking about a 4th factor between salts, carbonates, iron, and pH. And of course, we have varied mixtures of all of them up in these areas. And
Right,
generally
and
higher
leftover
pH.
nitrogen too. I mean, there's so many factors.
And the leftover nitrogen, how would that affect the plant?
I believe it just creates a, oh crap, it's before 8. I can't remember exactly at this very moment.
But leftover nitrogen would be beneficial or detrimental to the soybeans?
Detrimental from an IDC perspective.
Yeah. So basically tying up the iron or the iron calcium relationship. Next question. We got a couple minutes here yet. Any highlights about the mycorrhizal fungi? So basically that symbiotic relationship. So we basically know that it's magic glue, but is there anything that you want to tell us that can improve it? And you mentioned crop rotation or something the preceding crop. So any quick highlights on that part of it?
So I knew that that was going to be a question. And so I actually created a slide here to that kind of summarize some of the results here. So when mycorrhizal fungi were present, soybean biomass was highest at that neutral pH. Plants grown with those mycorrhizal fungi tended to have greener leaves with less IDC symptoms. One exception though was when grown with a high SCN population density. What they found was that adding mycorrhizal fungi actually tended to increase IDC symptoms. Now, there needs to be a little bit more work to see if this holds true, because they only, so when they had presence or absence of those mycorrhizal fungi, they had one specific fungal, just one species present. And so it's not just one species most likely present. There might be one more dominant in our individual fields. And so understanding how, you know, a mixture of these different fungal species and then individual fungal species, they really need to figure that out. One other thing that they found was that at that high SCN population density, the colonization of roots by the mycorrhizal fungi was actually reduced. And so I have those figures at the bottom. So 74.7% colonization at that high SCN population density versus 85.3% for zero. For the 0 SCN and 83.6% for 500 eggs per 100 cc's. And so if you have high levels of SCN, the theory behind it is that the plant roots are just so compromised and having such a difficult time that perhaps SCN gets there first. Or they figure that maybe mycorrhizal fungi, they're not exactly sure. So it's just all speculation.
Thanks, Angie. We need to wrap this up. So
but
the mycorrhizal fungi maybe sounds like a good crop update or blog of management practices that basically help or are detrimental to soybean production. But very interesting. And then last, can you show us your contact page once more? There we go. Thank you. So Angie Peltier, you can call her or look her up at the University of Minnesota Extension. She's based out of Crookston. There's her email and phone number. So thank you very much, Angie. We appreciate it.
Thank you very much, Chris. Good luck.
So on